Hi everyone, this is Nehad from Bio with Nehad channel. Today we're going to explain enzymes. We have four biological macromolecules and the proteins is one of them. We have been talking about the four biological macromolecules in the last four videos. Today we're gonna take one example of the proteins, which is enzymes, and we will be talking about the structure, function, and the mechanism of action. Let's get started. Starting with the enzymes function, you have to know that everything is done in your body. It has to be done through a series of chemical reactions, and those chemical reactions, they are controlled or like speeded up or catalyzed by enzymes. So enzymes, they are needed by all your chemical reactions. They are done in your body. And just to know that all the chemical reactions collectively that happen in your body, it is called metabolism. It can be divided into catabolism or anabolism. Anabolism, if you are uh, building up something, so you are putting or in your body the amino acids are put together to give the long polypeptide chain. So this is building up. It needs enzymes. This reaction needs enzyme to be catalyzed. Or it can be catabolism. Catabolism is breaking down something. So you break down food to get energy. And we studied that part before. So this is also catalyzed by the enzymes. Let me use this lovely comic just to show you the parts of the enzyme so I can explain to you later the mechanism of action of the enzyme. So the substrate, this is the substrate, and it's actually the molecule that the enzyme will be attached to or bonded with to start a chemical reaction. Either a big molecule in a catabolic reaction just to break it down into smaller molecules, and the enzyme will leave without any change. Or it may be small molecules, it will be attached to the enzyme, so the enzyme will just help or speed up. Only that one can just speed up attaching them together to give a big molecule, which is anabolic reaction. The most important part is the active site. This active site is the site that the enzyme can be attached to the substrate, like through it. And that active site has to be the same shape of the substrate, or the shape that fits the shape of the substrate. So if it's rectangular, the substrate has to be rectangular, or if it's a circular one, the substrate has to be circular one. And actually that leads us to talk more about the specificity of the enzyme. The enzyme is so specific. So this is the active site. So the substrate has to be the same shape, so it can fit that one, or it can be that shape, so it can fit the active site of that one, or it can be that shape. It's not. Actually, it's that shape somehow to fit that one. Okay, but it's specific because that one, it can't be attached to another enzyme, and that one cannot be attached to another enzyme. So each enzyme is a specific enzyme to that type of reaction. So we can say that the key is the substrate and the log is the enzyme. It's the log key complex. So here, this is how enzymes work. This is the enzyme and this is the substrate. It will just go, as we said, in the active site, and it has to be, of course, it has to fit that one, that active site. Then they will give the look key complex. Okay, it's only for like temporary, for like a while. Then it will release the products and leave the enzyme as it is. So this is that way, right? And it can be the other way as well. So that way it was like building up anabolism. Then we can have two bind here to the active site. They have to fit the active site. It gives the complex, the lock key complex. Then it will be together to give the opposite way. This is the product. So the reactants at the beginning and products at the end. If we start from that side, so the reactants will be that side and the products will be those side. So just to make it clear, look at that diagram. Basically it shows that it's a cycle. Okay, if we start from here, we have the two reactants, two substrate, they attach to the active site of the enzyme and they have to fit. Then it will give the enzyme substrate complex. The enzyme substrate, the substrate will be converted into the product. So later it will release the products and leave the enzyme as it was before. Then I'll change the color just to show you the different, the opposite side. This is, maybe we can just assume that this is the substrate or the reactant. Then it can go to the enzyme, the active site. Then it will give the temporary enzyme substrate complex, then enzyme product complex, and release the two products. So it's like a cycle. Synthesis is two or like small units, small molecules it will give after the reaction, after the whole process. It will give a big product, or breaking down, so the big product can be broken down by a reaction into small ones. And if you remember, it's catabolism and anabolism. Catabolism breaking down, anabolism building up. Just for more illustration, this is, I'll start from here. So this is the substrate, then broken up into the two products, and it will give two products. Question time. This is metabolism or catabolism? This is synthesis or breaking down? Okay, if you say that it's building up or anabolism, it's wrong. So the right answer is breaking down, and you can say that it's a synthesis reaction. And to start any reaction, you have to have a certain amount of energy just to start it. It is called activation energy. To start any reaction, you have to have this minimum energy to start. Okay, so the activation energy, the energy needed to start a reaction. And I feel it's so high here, just to have all of this to start the reaction. Actually, this reaction is, this part is the reactants and this part it's the products. Okay, so the reactants here, they are OH and CH3Cl. Then they made the temporary complex, then they gave the products. What are the products? We switched. We have here as products CH3OH, and we have the Cl here alone. Okay, so what is like the effect of enzymes if we have an enzyme in this reaction? Think about it. Actually, the activation energy, this is the one without enzymes, and this is the one with enzymes. So it lowers the activation energy. It lowers the energy that it's needed to start a reaction. So it's way easier to start this reaction. That's why it speeds up, or that's how it speeds up actually the reaction. So this is the energy amount that it's needed by this reaction without an enzyme. So it's so high, but this is the amount of energy with the enzyme to start the reaction. Okay, so this energy, it's needed to start the reaction, activation energy. This is with the enzyme, it's so less. Now we're going to start the factors affect the enzyme activity. Number one is temperature. Then we're going to explain the pH and substrate concentration. The temperature, when you give more energy, when you raise the temperature or giving more thermal energy to the molecules, or the molecules that like the substrate molecules, so they will be having more kinetic energy, so they will move more, and the probability of colliding to the active sites, it will be more, so the activity or the rate of reaction will be more. Okay, so the temperature will be increasing till it reach the peak, or you can name it optimal temperature. So the optimal temperature, it's the temperature that the enzyme will best perform at. Okay, so it will just be the fastest rate of reaction. Then it will start to decrease. Just think why. So let me help you. Here the collisions, by more temperature, the movement of the substrate will be more, the collisions will be more, and the probability of the substrate like enters the active site will be more. And more temperature, so what happens? So more temperature will here start to decrease the rate of reaction. Why? Because the enzyme will start, like higher temperature, the enzyme is a protein, so the protein will start to break down, so the shape of protein will change and the active site shape will change. So maybe it will be that shape, so it will not fit the enzyme anymore, and the enzyme will stop functioning. So it will decrease till it will stop completely. The rate of reaction will be 0. The point is, this denaturation is irreversible. So when the protein is denatured and it will be broken down, nothing will give it the same shape again. It will not be able to build up and give the same shape again. So the denaturation is a irreversible process. Question time. If you have this graph in your exam and they asked you what is the optimal temperature for that enzyme, of that reaction specifically, so you will go to the peak, so the highest point, and you're going to go down, and it's actually before 40, maybe 38 or something. The second factor that affects the enzyme activity is pH. You know the pH, the 7, it's the neutral. The less you go, the more acidic you have, and the more you go, the more basic you have. And the enzyme, each enzyme, same as temperature, it has an optimal or ideal pH to function, the maximum performance. Okay, so this one, for example, the rate of reaction, that side, and on x-axis it's the pH. Then the more you go up, the more pH, the activity will be more, the rate of reaction will be like faster, more. And then till the peak, it's the maximum, the optimal pH. Then it will start to go down, the activity will be less, so the pH is not the, the best. Like it will not perform at this pH, it will not perform the best. It will be less till it denaturates. What the meaning of denaturating? The same as temperature, that the denaturating, the shape of enzyme, the active site, it will not be the same, it will change, so the enzyme will not function, because actually the enzyme shape will not fit the active site shape. Okay, for example, in your stomach, it's very acidic, so the enzyme that works in your stomach, the optimal pH for that enzyme must be very acidic, otherwise it will not work. Okay, so to break down the proteins inside your stomach, for example, you need the acidic pH. That's why the stomach has this acidic pH. Question time. What is the optimal pH of that enzyme? So you will go, as we said, to the peak and go down. So the pH of that enzyme, that question, it's 8. Guess what is the third factor that affects the enzyme activity, the rate of reaction during the activity presence? This is the amount of the substrate. So if I have this amount of substrate, actually it's 1, and this or higher concentration of the substrate, the enzyme activity will be more for each of them. Of course, that one. If you said number 2, so this is the right answer. The third factor that affects the enzyme activity or the rate of reaction of that enzyme is the concentration of the substrate. The more substrate, the higher rate of reaction. Okay, so the higher activity. So when the substrate amount is more, there is more probability that the active sites will be occupied by substrates, and the reaction will be, so the rate of reaction will be more. So more substrate, the more rate of reaction, till all the active sites of all the enzymes are already occupied by the substrates. More substrates, it doesn't affect, because no more active sites, no more enzymes, so it will be like somehow constant. It will not go for more activity, because no more enzymes. So at that level, all the enzymes are, the active sites are already occupied by their substrates, no free active sites, so it will just stay constant, no more room for substrates. So just now tell me the difference between the normal enzyme and the denaturated enzyme. Yeah, the active site of that enzyme, the normal enzyme, it fits its substrate, but for the denaturated one, it doesn't fit the substrate. The shape of the active site is changed, so it doesn't fit the substrate anymore, so it will not function, it will not work. For most of the enzymes in your body, the optimal temperature ranges from 35 till 40 degrees, so around your body temperature. And for the pH, the optimal pH is close to the neutral one, but we have many acidic, like the stomach one, we have also many basic. The suffix ase, it's for all the enzymes. So we have amylase, we have protease for the protein, amylase for the starch breaking down. What else we have? We have for the protein, for the starch, we have for the lipase, we have lipase for the lipids or the fats digesting. So the digestive system, it has many enzymes to help it like does its function. Now we're going to explain how the enzyme activity can be decreased or stopped by a chemical called inhibitor. So the inhibitor is a chemical that interferes with an enzyme's activity. We have two types of inhibitors. We have a competitive inhibitor and a non-competitive inhibitor. Actually, this is the normal active site, the normal substrate and enzyme. So this is the normal relation somehow. But here we have something that blocks the active site. So it prevents the substrate from combining to the enzyme, and this is the competitive inhibitor. The non-competitive inhibitor, it's the inhibitor will be like bind to somewhere else. It's not the active site, but any other place other than the active site, change the shape of the active site, and it stops the substrate from binding to the active site. So either blocks the active site or changing the shape of active site to stop the or decrease the activity of the enzyme. Actually, the exact opposite of the inhibitors, it's the cofactors and the coenzymes. They are non-protein molecules. They help the enzymes to perform their work. The cofactors, they are ions or metal atoms, like maybe iron ions, and we have also the coenzymes, which is like the vitamins. Thank you for watching. Wait for the next video, subscribe and like. Thank you.